DNA stands for deoxyribonucleic acid. So it's a nucleic acid. And DNA is the chemical compound that contains our genetics. So when we talk about genes, genetics, genomes, we're talking about DNA. And the DNA actually carries the genetic instructions in order to make you who you are. So a gene is a segment of DNA that codes for a protein. So you might have a gene that says you are going to make the protein melanin, which gives you your skin color. And if you have a gene that says you're going to make more melanin, you're going to have a darker skin color. If your gene says less melanin, then you're going to have a lighter skin color. These proteins also apply to enzymes, meaning which different chemical reactions you can catalyze, whether you're going to have a genetic disease or not, which diseases you're resistant to, as well as your physical appearance. And if we look at, we call this the central dogma of biology, and that's this here, that DNA, which contains the genes, gets converted to RNA, which then gets converted to a protein. And we'll talk about how this happens a little bit later when we talk about protein synthesis. But for now, we're talking about this material that eventually we're going to be able to talk about how that translates into a protein. So DNA is a polymer. As we mentioned, it is a type of nucleic acid, and nucleic acids were one of the macromolecules that we talked about at the very beginning of this semester. So a polymer is made up of monomers, and in DNA, the monomers are nucleotides. And if we look at this picture here, this is a picture of a kind of a generic nucleotide. So all nucleotides have a 5-carbon sugar called deoxyribose, a phosphate group, and a base that contains nitrogen. And we call this a nitrogenous base or just the base. Now that base component, so if we go back a slide here, this base component here is what changes between the different nucleotides. So you may have heard of adenine, thymine, cytosine, and guanine. That's where this nitrogenous base comes into play. So if we go back to this slide, there are 4 different types of nitrogenous bases, and they all have a different chemical formula. So first let's take a look at here. We've just got our deoxyriboses here and our phosphates here. We have the same thing over here with the deoxyriboses and phosphates. And so the bases are either a double ring structure here or a single ring structure here. So thymine, this is the thymine base that exists on a thymine nucleotide. This is the guanine base that is attached to the sugar deoxyribose and the phosphate to make a guanine nucleotide. Cytosine is also a single ring, although it does differ in the arrangement of the atoms from thymine. So even though it's the same size, it does have some differences to it. And then adenine is similar to guanine, and then it has 2 rings that make up the nitrogen base part of it. But just like thymine and cytosine are still slightly different with regards to the arrangement of atoms, so are adenine and guanine. So the double ring structure molecules are called purines. So guanine and adenine are purines. And then pyrimidines are the single ring structures, which include thymine and cytosine. Now if we look at, this is kind of a cartoony drawing of a DNA molecule, and you'll notice that it's the same width all the way across the double helix. Now this gave early scientists a clue to indicate that a purine always had to pair with a pyrimidine, because if a purine bonded with another purine, it would make part of the DNA double helix wider. And then if, say, thymine and cytosine bonded together, it would make that part of the DNA double helix kind of thinner in that area. And we just don't see that. So we know that a purine always pairs with a pyrimidine. And we now know that adenine always bonds with thymine, and guanine always binds with cytosine. Now, one way to remember base pairing is to use this little mnemonic here. So first of all, let's take a look at the GCAT. GCAT shows you that G always pairs with C and A always pairs with T. All right, so GCAT is guanine always pairs with cytosine, adenine always pairs with thymine. And then the puppy comes into play because guanine is a purine, which starts with PU. Cytosine is a pyrimidine, which starts with PY. Adenine is a purine, which starts with PU. And thymine is a pyrimidine, which starts with PY. So once again, GCAT means that guanine always binds with cytosine, and adenine always pairs up with thymine. And then guanine is a purine, cytosine is a pyrimidine, adenine is a purine, and thymine is a pyrimidine. Now, if we take a look at the overall structure of DNA, it is a double helix. So imagine a ladder that has been twisted around itself. The deoxyribose molecules and the phosphates make up the backbone. So this is this kind of blue strip on the outside here. And you can see them here. So this is a deoxyribose, 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 phosphate, 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 phosphate. So the phosphate connects one deoxyribose to the next deoxyribose. The rungs of the ladder, so the middle part of it, is where the bases come into place. So you can see that we've got the thymine nitrogen-containing base pairing up with adenine. Guanine is pairing up with cytosine. Cytosine pairs up with guanine, and adenine pairs up with thymine. And this structure was really discovered back in the 1950s, so that's not that long ago that we kind of figured out what these molecules look like, which is pretty impressive given how much we know about genetics now. Now the nucleotide backbone, so remember we've got the deoxyribose and a phosphate. These molecules are bonded together by covalent bonds. And if you recall, covalent bonds are fairly strong bonds. So it's pretty hard to break a covalent bond. It can be done, but it takes a lot of energy and probably some enzymes to help out as well. The bases are paired together using hydrogen bonds. And hydrogen bonds are significantly weaker than covalent bonds. So this kind of comes into play when DNA is replicated, because during DNA replication, the bonds between the bases get broken to open up the two sides of the DNA molecule, but the covalent bonds, for the most part, stay intact. Another thing we've got to look at is that deoxyribose is a 5-carbon sugar. So you see that it is kind of drawn like a pentagon here. And you'll notice these little numbers here. So there's 1', 2', 3', 4', and 5'. So that little apostrophe is read as prime. And this is just a way to kind of number the carbons, because you're going to notice that on one end of the DNA, you've got a 5' carbon, at least on this side, it's the one that is on this end. And then the 3' carbon, which is the one that connects to the phosphate group, is on this side. Now, on the opposite strand, it's going the opposite direction. So notice that strands always have, or one strand, sorry, I keep hitting the wrong button. One strand runs from 5' to 3'. The other one goes from 5' to 3', but in the opposite direction. And you can kind of see where the pentagons are pointing. So these are pointing down, and these are pointing up. So it kind of shows you how they're running in opposite directions. So we call this anti-parallel. Anti-parallel means that, well, first of all, parallel means two lines that are kind of running next to each other that never intersect with each other. Anti means that they're running in opposite directions. So you can kind of think of it as like a two-way street, where this street is going in this direction, and this lane, I should say, is going in this direction. Now, the reason why we're mentioning this here is because when we talk about DNA replication, the direction of the strand matters. And so this is going to come back up in kind of our next video as we talk about DNA structure, or I'm sorry, DNA replication. So we've introduced that DNA makes up your genetics, it's your genetic code. So maybe you've also heard about a term called the genome. So back in the late 90s, early 2000s, there's a lot of work being done on the Human Genome Project. And this was the first time scientists ever sat down and mapped out the base pair sequence of an entire human genome. So a genome is just an organism's complete set of DNA. So all of the DNA in each of your cells that codes for you is your genome. So we've talked a little bit about how in eukaryotes we have 46 total chromosomes that are packaged as 23 pairs. And these are the chromosomes that we get from our parents. And so these 23 pairs of chromosomes make up your genome. And each cell contains an entire copy of the genome with some exceptions, but most cells in your body have the entire set of chromosomes that you have in all your other cells. Humans have about 3 billion base pairs in each cell. That's a lot. So it's pretty impressive how many bases we have that make up our genome. That's a lot of information to code for you. And each chromosome contains hundreds to thousands of genes. So lots and lots of instructions or recipes, if you want to think of it that way, for proteins. Now, prokaryotic DNA is packaged in a single circular chromosome, and it tends to be significantly smaller. So imagine we've got a long strand of DNA made up of 3 billion base pairs. That could get tangled up really easily. So imagine if you've got your earbuds and you throw them in your backpack or your purse, and when you go to pull them out, they're all tangled around each other, and you've got to spend all this time untangling it just so you can listen to your music. So imagine if the DNA was just thrown in the nucleus of the cell and it had to be kind of untangled every time we wanted to make another copy of DNA or go through DNA replication. So the first thing is that the strands of DNA are wrapped around proteins called histones. So this is kind of like I have this little plastic thing that I can wrap my earbuds around, so when I put them in my purse they don't get tangled up. And this unit right here, so a histone and the DNA wrapped around it, is called a nucleosome. Now, several nucleosomes with DNA wrapped around them get stuck together kind of like a beaded necklace. All right, so each one of these units with the DNA and then the histone here is a nucleosome, and all together this makes up chromatin. And then just prior to DNA replication, the chromatin condenses and it winds even more around itself in order to form a chromosome. And a chromosome is visible under a compound microscope, whereas these other structures really aren't. We can kind of see when a stain picks up the chromatin, we can kind of see where the DNA is, but the chromosomes are distinct and we can actually see them pretty clearly underneath a compound microscope.